Hello. Today we're going to look at the 16th lecture in the series of biochemistry talks from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we're going to talk about nucleotides as signals and also nucleotides as functioning elements in the biochemical system. So to reiterate, these are the informational repository in the biological organism in the more permanent form of DNA, and RNA has a more flexible role going in both directions, both towards DNA and down to protein. So again, nucleotides can function in many capacities, and one of these is a signal where ATP is released at synapses. The membrane changes in potential in the receptor cell. Nucleic acid technology, we talk about the genome. The genome, by definition, is a complete haploid genetic complement, and you can see these in chloroplasts and then also in mitochondria, which are derived from bacteria, evolutionarily speaking. A DNA clone is an identical copy produced through amplification. So to get a DNA clone, we talk about restriction endonucleases. These will recognize specific structural sequences in DNA and cleave them in specific sites. And these separate fragments are synthesized by polymerase chain reaction, which we reviewed in our last talk. You can select a small DNA molecule that can replicate autonomously. You join the DNA cloning vector to the DNA segment, which is to be amplified, and this forms recombinant DNA. The recombinant DNA is then transferred into a host organism, and you can select the host cells which contain recombinant DNA. E. coli is the most common host cell. The DNA vectors that are usually used, the DNA cloning vector, usually those are bat plasmids and bacteriophages, so you can get amplification of a specific DNA sequence that you can determine in the host organism, isolate that segment, cleave it to a DNA cloning vector, and it would be part of the E. coli, and then replicated from that. So if we look, for example, we have a cloning vector, which is a plasmid, a eukaryotic chromosome, which is digested, fragments the one of interest, is then combined with a restriction endonuclease cleave component, and this is a recombinant vector here, which is introduced into the host cell as host DNA, and the replication of the cell occurs, and you get replication of the plasmid vector with the recombinant DNA. So this is the part you're interested in. It's amplified if there's a marker attached or a biological activity that is consistent or characteristic of this segment of interest. You can isolate it and then go ahead and get recombinant. So this is the kind of process you get with a polymerase chain reaction to get more entities. There are two enzyme classes that are used in the recombinant DNA. Restriction endonuclease is called a restriction enzyme. Those generate the small fragments. You can get three types of these restriction endonucleases from bacteria. Take the one off the shelf that works for you. A DNA ligase will join the vector, whether it's a plasmid or some other form of DNA, to the fragment. You get the recombinant DNA from that. The products depend on the mode of action and the number of site-specific sequences available. You can stop the reaction to get a partial digest, and those DNA fragments can be purified to find out where you are in the process. You can also isolate the fragment of interest, join it to the vector with the ligase, and then add DNA to facilitate binding. When you're dealing with these restriction endonucleases, you can have two strands of DNA. You can cut blunt ends, which means you cut both strands in the same location. Both strands are cut. Or you can get sticky ends where they cut off center and they are cutting at different distances in the opposing strands. For the vectors, you can use plasmids, circular DNA fragments, which have bacterial origin, and these will interact with the main bacterial genome, usually an E. coli species. Method of conferring resistance or performing new cell functions occurs. These are ways you can select out the component of interest. You can employ resistant bacterial forms or not, and that can be a characteristic of the clone that you're trying to isolate and characterize. If you have the same replication origin, you don't want different plasmids. Those will give incomplete mixed results. You want a pure plasmid species and introduce that with the DNA strand of interest into the host bacterium. And you can incubate it. You can also have a host increase, or you can increase the voltage. The most successful plasmid uptake occurs by linking it to a fluorescent dye marker or any entity that retains cell viability. If you want longer segment introductions, you don't really use E. coli. You can use yeast as a host cell, and you can generate longer segments with electrophoresis. And amplified genes can be utilized. Those can produce proteins in bulk. You cannot really use eukaryotic sequences. They don't really function in bacterial hosts unless you additionally add transcription translation factors. So these factor coding sequences must be in proximity. So what you want to have is leader and trailer sequences to the nucleic acid fragment. These would be recognized by a eukaryote because these are more complex, and those can then invoke the biological activity. You can use numerous host systems for the production or recombination of recombinant proteins. Use bacteria, yeast. You can even use insects infected with viruses and mammalian cells in culture. One element that's used commonly is to use adenovirus and retroviruses as vectors. These would be combined with DNA sequences of interest, and you can get mass amounts of product using that as the vector. Altered genes get altered proteins. You can have oligonucleotide-directed mutagenesis. You can denature a gene. You can separate the strands, anneal the complementary strands with an altered gene sequence, and then re-anneal those strands that you produce. You can amplify them with PCR. You can select the desired strand for mutation and then introduce into a plasmid and then to a cell. So you denature, separate, anneal, have the altered sequence included, re-anneal, amplify, select that desired strand, and then put it in a plasmid and then into the cell. So this is an example here. Site-directed mutagenesis, a recombinant vector here. You cleave it with a restriction endonuclease. You insert a synthetic DNA sequence with the mutation. That's site-directed. There's also oligonucleotide directed, where you have the gene, the target site for mutation in the plasmid. You put in primers, so this is done in a more simplified system that you want to make the entity more complex. So you denature the plasmid, anneal the oligonucleotide primer with the mutation. So you have annealing right here. Use DNA polymerase to extend and incorporate the mutagenic primers. So you've got this the primer here, and then you extend the synthesis, so you get the double ring. Then you can digest the non-mutated template. You can use methylation-specific nuclease and re-anneal these newly synthesized strands, and then you can transform the double-strand DNA, put it into cells, and the cell repairs will have nicks in the mutated plasmid. So these are two ways of doing it with site-specific mutagenesis and oligonucleotide-directed mutagenesis. A lot of this has been replaced by CRISPR. We'll be spending some time on it. Fusion protein methodology, you remove the intervening DNA sequence, separate in two segments which code for separate proteins. One protein is produced, then you can add a recognition tag to a protein for purification or bind it for affinity chromatography. So producing a small protein with a marker on is used, and you can have the marker guide if you've got the correct mutation sequence in question. You can also use a reverse transcriptase to produce DNA from an RNA template. Oftentimes this will be done to have a DNA template, and the DNA template can mass produce portions of RNA for a variety of biological and medical purposes. DNA libraries can be formed. You have a collection of DNA clones or fragments that are derived from DNA digestion, amplification, and these are a vast array of reagents that you can produce. You can have reagents that are comprised of various segments, various oligonucleotides, and you can use those right off the shelf and produce a lot of these primers, a lot of the markers, a lot of leader and trailer sequences to get the desired result. Comparative genomics, you have characterization of biological function through DNA sequence homologies. This is analogous to amino acid structure and related protein function. So comparative genomics looks at DNA sequences over a variety of organisms, whether same species or interspecies comparisons. There's also fusion proteins. This is a marker protein. It can be covalently attached to the structure of interest. I've addressed this just a few minutes ago. There's a fluorescent marker. It could be a chemiluminescent marker. And you can visualize this and quantitate it. And you can determine the location in the cell as well as hybrid markers, which will produce a given outcome of a coloration. If you've got proximity of the two individual markers together, they will form a different signal, and that would cause a combination of genetic structural perturbations. So one example of this is a green fluorescent protein. You can introduce a gene, a GFP gene, introduce a host gene. They're both translated into protein, which bears that marker. The GFP can be cloned. It has no effect on the host protein function, but it can persist and not denature. So this can be a continuing marker that you can use throughout the course of an experiment to determine if you have preservation and function of the moiety to which this GFP gene is attached. We talk about DNA microarrays. This is simultaneous and rapid screening of thousands of genes. You can take a DNA sequence, you can convert it into shorter segments, and you can then synthesize independent sequences on a small solid surface, such as a chip. And once formed, that microarray of sequences can be paired with other DNA or RNA sources to determine sequence homology. So you can have a solid matrix with DNA sequences. You can produce a homogenate of DNA sequences to find out if you have attachment or recognition of a specific DNA sequence to your template, which has a certain characteristic. You can also pair this with fluorescent labeled nucleotides or two different probes to determine combined genomes. You can assess conditions for expression in a variety of conditions. So one example is to take a fertilized egg and you isolate messenger RNAs, and each RNA sample expresses all the genes at that stage. So you have fertilized egg, you have a tadpole with its array of messenger RNAs, and you can convert these with traverse transcriptase. You can produce the complementary DNAs from this using fluorescent labeled deoxyribonucleoside triphosphate. So these are labeled from the fertilized egg, labeled from the tadpole, and you can have a DNA microarray of fluorescent DNAs. They anneal to the complementary sequences, and the chemiluminescent or fluorescent chip gives a color that you can see very readily on the microarray just by simply applying that mixture to it. You can find out if you have individual results or combined sequence homology by determining the expression based on the color. So higher expression of the tadpole, for example, here, you can have higher expression in the single cell stage of green. You can have unchanged expression yellow, and these are very useful because the combination of colors will give a color that reflects the combination or lack thereof. CRISPR is very hot today. It stands for clustered, regularly interspersed, short palindromic repeats. You target a gene, you can remove it, or remove it, insert a different gene. There's an endonuclease from bacteria that has a sequence-specific recognition and cleavage site. This deletion of DNA is subsequently repaired through homologous recombination. So the CRISPR, evolutionarily speaking, is a bacterial immune system. It protects against bacteriophages. CRISPR sequences are embedded in the bacterial host genome. That genome is adjacent to a phage sequence, so a bacteria gets infected by a phage. The phage sequence is incorporated into the bacterial genome and, for all intents and purposes, constitutes a library against which the bacteria can mount an immune response if it recounters the phage again. So the immune response is elicited, the CRISPR and the phage sequences are excised from the host. You encounter the phage nucleic acid in circulation. The complementary sequence binding occurs between the extraneous phage genome and that derived from the host that was in storage. You get a nuclease that is coated and it cleaves and destroys the phage DNA. By using this specific sequence, you can use this in biological systems. So what we're looking at then is, for example, the CRISPR sequence here. This is a recombination fragment that's attached. It goes from the cytosol into the nucleus. So this CRISPR plasmid with the recombination fragment goes in here, and this is critical. But you've got the host, the genome. It separates, and you get a target that is present here. It's a chosen target sequence, which is a part and parcel of this recombination fragment. It will recognize a part of the gene that is of interest. It will complement and bind close to the gene of sequence, and there will be a nuclease which will excise this part. You can excise it and then it will reanneal, which means a deletion, or you can have a lot of DNA nucleotide in the area and it can recomplement and cause a corrected version in this point here. So you get a double-stranded break at this point with opening here, and you can fill it in with the corrected version. You can insert a recombination fragment here with the same process. You can remove it or you can insert. And the critical thing is that the complementary segment on the DNA has to be close to the target that you want to remove. If the target that you want to remove and the complementary site is far away, then you can get more errors. So you want these two close together. So a lot of times when you introduce one of these CRISPR sequences, you have to make sure that the sequence that the CRISPR acts on and breaks is present close to the target that you're interested in. And that's a mathematical construct. One advantage of the CRISPR sequence that is complementary to the host is is that there are oftentimes several of these. So it's a function of finding the one that has the genetic sequence that you're interested in either removing, deleting, or deleting and replacing, which is the repair component here. This deletion, that's repair. CRISPR can be used in either component. There's a variety of bacteria which have CRISPR technology within them. Streptococcus pyogenes is one particular strain of that, which has a very limited, a very elegant single-strand guide DNA. So the guide DNA is what you're using, the guide RNA, I should say, is what's used to recognize the invading phage sequence. And you can adapt the system to plasmid vectors, and you can extend utility to a variety of different systems. So you can either excise or you can excise and insert processes. Again, the difference between the two is that the insertion requires a synthetic component to that, so that's a little bit more complicated, but you can remove one gene, put in another. You can have, if you just simply do an excision, you can have basically a deletion mutation. You want to avoid frame shift mutations, which cause a lot of havoc and basically end the sequence at the point of repair. The Human Genome Project was is a component of all this. The draft form was completed in 2001, the final form in 2004. Numerous species and extinct forms have had this work done. You can compare the DNA sequences and allow for assessment of function. The uncharacterized component DNA is about 40%. Their function is unknown. They may represent archival evolutionary inactive forms, sort of like Neanderthal DNA. You know, it's there. And it has no particular function. It may be protective against nucleases, but for the most part, it's unknown and uncharacterized. So today we have about 100,000 genes with 3.2 billion base pairs and 20,000 protein coding genes. Fruit flies, by contrast, have 13,600 protein coding genes. Rice plants have 38,000 protein coding genes. There are introns, which are non-translated DNA sequences, and exons, which are the coded segments. You can splice the introns from a DNA sequence and produce a contiguous protein product. Exons are usually a contiguous translated segment of a larger protein. You can have splicing occurring. It's not always uniform, so you get more complex structures. There are only a few introns in bacteria. Mainly you see introns in mammalian systems. They have more DNA. There's more opportunity for those DNA sequences to be translated or non-transcribed. So in humans, 1.5% DNA codes for exons and 30% code as introns. So when we talk about these messenger RNA transcripts, we again have the 5' and the 3' areas with introns and exons, non-transcribed, transcribed areas that are produced. And you can have splicing of introns, so you remove these, and then you have contiguous splice messenger RNA comprised solely of the translated intermediates. Whether these are functional is another matter. Sometimes if there's no function attached to these introns, then it's acceptable to have this sort of approximation of these various coding regions on this open reading frame. Transposons are DNA segments which move from one location to another within the genome. They can be DNA or RNA, the latter of which has reverse transcriptase necessary to get to DNA from the RNA substrate. It can constitute 50% of human DNA, but most of these are inactive. There are also short sequence repeats, which are DNA sequences which can repeat millions of times. These are seen in centromeres and telomeres. during mitosis. There are short nucleotide polymorphisms. These are small single base variations, about 1 per 1,000 base pairs, and they govern genetic differences between individuals. So for inheritance, we talk about small nucleotide polymorphisms. They mix and match. A grouping of these polymorphisms is a haplotype, and a set of these haplotypes is inherited together. So the amount of interspecies variance is only 1.23% between humans and chimpanzees, 0.1% variance between humans. The number of transposons, insertions, and deletions, however, differ markedly between species. So these polymorphisms can produce a lot of similarities between species, but the internal base structure is quite different and lends us or different characteristics. So you can have areas of accelerated divergence between species and areas of genome that have great differences in the primary sequence. For linkage analysis, a study of species sequences to ascertain specific diseases, one gene or multi-linked. So a lot of diseases are multifactorial, some such as sickle cell anemia or a single point mutation in a valine moiety in hemoglobin. You also have single nucleotide polymorphisms. These are a complicating factor, and you're looking again at the base sequence near the affected gene, not within the gene. You're looking for consistent structural characteristics in affected individuals. So you can track population migration through history. This has been done to determine whether you have contiguous populations that have been separated over time by water bodies or some other element where they moved and water covered the connecting areas. Neanderthals, for example, are extinct. There was a hybrid with Homo sapiens, but they could not reproduce well. And there was a hypothesis that there was a link to autoimmune disease, and everybody has a little bit of Neanderthal DNA in them. And with that, we will stop. Thank you very much.